Hello students, I am Sonali Kelkar from City International School, and today we are going to do the chapter Heredity and Evolution. In this chapter we are going to do the dihybrid cross and sex determination, and hence we are going to complete this chapter. Okay, so let's start today's session. Let's start with the recap. Okay, we have done in the last session about the monohybrid cross, right? Now what do we understand by monohybrid cross? Mono is a single, okay, single character, right? Here we are taking one single contrast character, and we have taken here as tall capital T and the short character which is denoted by the lowercase of t. Okay, so capital T and small t, these are the one set of contrast character. And you can see in the other picture we have taken the purple and the white flower. Purple is a dominant and white is recessive. So this is one set of contrast character. In this monohybrid cross, first of all, the parents which are pure in nature. What do we mean by pure? That means both the genes are similar, that is they are homozygous in nature, right? Both the parents, one is the homozygous dominant and the other one is homozygous recessive. Both are pure, it means both have the same type of set of genes, right? First of all, they are crossed with each other. So when they have crossed with each other in the first filial generation, here we saw that all the plants were tall. All the plants were tall. And why these plants were tall? Because we got the genotype. If we see the genotype over here, we have got the heterozygous genes over here, right? Heterozygous genes where one is the dominant, the other is the recessive genes, right? So whenever there are two contrast characters, the one which is dominant will show its influence, right? And this is somewhat is called law of dominance. Okay, the gene which is dominant in nature will show its influence in the progeny, right? So Mendel came to the conclusion in the first generation that all the plants were tall. So then he did the self-fertilization of these two plants, and what did he observe? Okay, after doing the self-fertilization of these plants, he got three tall plants and one recessive plant. Okay, so three tall plants and one recessive plant, because these are the observable things, so we call them phenotype. Phenotype means the physical appearance of a progeny, that is called phenotype. And genotype, that is he got one homozygous dominant, two heterozygous dominant and one homozygous recessive genes, right? So genotype is the genetical constituent of the plants, right? And that is 1, okay, is to 2 is to 1. Clear? So this is what was a result of the monohybrid cross. What was the observation? We have already discussed about the observation, right children? Law of dominance, as I've already discussed you, what is the law of dominance? When parents having pure contrasting characters are crossed, then only one character express itself in the F2 generation, right? We saw that one character could able to express itself in the second generation, right? So the character which expressed itself is called dominant character, and the other which couldn't able to express itself is a recessive character, right? Now what do we mean by law of segregation? It's very simple. See, here we have taken the heterozygous female parent, right, and heterozygous male parent, right? In the F2 generation, in the F2 generation, Mendel self-fertilized the plants. Okay, what was a genotype of F2 generation? That is capital T and small t, right? So this is the genotype of a parent. This is the genotype of a parent, right? And let's take this parent is a male parent, so I'm going to represent it with this sign. And what about the genotype of female parent? That is capital T and small t, right? And it will be represented by this sign. Fine, by this sign. Now these parents, definitely the parents will form gametes. What are gametes? These are the germ cells or the sex cells, right? And the parent, which is male, will form sperms, and we know that in sperms there are n number of chromosomes. Okay, don't consider it. There are n number of chromosomes, that is half the number of chromosomes, right? So in sperms only half the chromosomes are getting in, right? So each character that of the tall each character of a trait over here is tall and short trait, will go separately in the gametes. So let's take the first gamete, will get the T, and the other gamete will get the small t, right? So these characters these traits are dividing independently, and they are going to a different gametes, right? So this segregation of the characters separately into different gametes is known as law of segregation, right? Here also the female gamete will produce the capital T and the small t, right? So this is the female gamete. Fine. So we have done the crossing of these two gametes, right? Okay, so this is all about the law of segregation. Here you can see the female parent. Okay, while the formation of the gametes, the traits are getting divided and they are getting separated and they get entered into the gametes. Okay, here also the sperm, one of the sperm is having the gene which is carrying a tall gene, and the other gamete which is carrying the short gene. Okay, so this is how the gametes are formed and the characters are distributed while the formation of gametes, or the genes are distributed while the formation of gametes, right? Here we will see the definition: member of a pair of homologous chromosomes separate during the formation of gametes and are distributed to different gametes so that every gamete receive only one member of a pair. Now coming to the next cross, that is dihybrid cross, right? Now what do we mean by di? Di means two. Hybrid is just opposite of pure, as I told you. That means hybrid is a heterozygous condition of gamete, heterozygous condition of a cell, right? It is a cross between the two plants which have two pairs of contrasting character. In monohybrid, we have taken only one pair of contrasting character. Okay, that is tall and short. But over here, we are going to take two pairs of contrasting character. Okay, so the two pairs of contrasting characters that we are going to take here is round, which is represented by the capital R. And why it is represented by capital R? Because round is a dominant character. And yellow, which is represented by the capital Y. Okay, capital Y means yellow is a dominant character. So round and yellow too are the dominant character, right? And our green is a recessive character which is represented by small r. Clear? And wrinkle is a recessive character. Wrinkle is a recessive character which is represented by small r, and green is a recessive character which is represented by small y. Okay, so here we have taken the two contrast characters, that is a round and wrinkle, yellow and green. Here let's do the crossover first. In the parent generation, Mendel did the crossing of the parent generation, right? So it was a cross fertilization. In the first he did cross fertilization. I'll just write cross F, that is fertilization. Okay, cross fertilization, he took parents male and female from a different having a different characters, and then he took, we know that it is dihybrid cross, so he took the two contrast character, and that belongs to the two different plants, right? And that is why we call that it is a cross fertilization, right? Here he took the one plant having the round and green character, and the other plant which has wrinkle and yellow characters, right? So parents having these two characters. As we know that while forming the gametes, these characters separately enters into the gametes, right? So in the first case, that is capital R and capital R, small y and small y, right? This is the parent, and while forming the gametes, these character will go separately. So capital R will come in this, and small y will form other gamete. So okay, will come into this gamete. If I do this with the others also, let's take, if I do with the this R, initially I took this R and this Y, and this R and this Y. If you see, all the gametes are of the same genotype. All the gametes are round and green. All the gametes have the same genotype, that is round and green. Fine. So these are the gamete. That is why if you see here, we have taken the gamete R and small y. Okay, we took the only one gamete. And what about the other set of the gamete formed by the different parent? Okay, here also the gametes, when they are forming, we have to take one, okay, gene responsible for the one character, that is R is wrinkle and Y is yellow. So I will get the gametes having the same genotype. Okay, they all are wrinkle and yellow, wrinkle and yellow, and here also wrinkle and yellow. So we know that in the process of meiosis, meiosis is a division that takes place in reproductive cells. In that process, four gametes are formed. Okay, so you can say that these all are egg or these are sperm. Okay, we are just taking it hypothetically. It's just a hypothetical. There is no such experimental based over here, right? So we have seen that the segregation of the two hypothetical genes located on different chromosomes, right? So we got our gametes, right? So these gametes are later on fertilized with each other, right? And what do we get when these gametes, that is capital R and small y, and small r and capital Y, okay, the gametes coming from the different parents, when we fertilized them in the first generation, in the first filial generation, okay, the progeny that I am getting will have the character that is round and yellow, because capital R is present over here. Okay, and we have to rearrange this. So I have to take capital R and small r over here, capital R and small r, that I'm going to keep these two gametes together, and capital Y and small y. Okay, the identical or alleles I'm keeping together. One allele responsible, capital R, responsible for round, small r responsible for wrinkle, yellow, capital Y responsible for the yellow, and small y responsible for the green color, right? So what will be the phenotype? Phenotype, that is physical appearance, because we have a R that is round, which is dominant, and yellow, which is also dominant, right? So dominant character is going to express themselves in the progeny, right, according to the law of dominance. So I am getting all the plants which have round and yellow seeds, right? What about the seeds? Round and yellow seeds, right? So this is the result of F1 generation, right? All the seeds were round and yellow. Now coming to the F2 generation, right? So here we got our progeny R, which has the genotype capital R, small r, round, and capital Y, small y, which stands for yellow, right? So round and yellow, heterozygous condition, of course, right? And in the first filial generation, regarding the parents, we did the cross pollination or the crossover. But in the next F2 generation, what we are going to do, we are going to self-pollinate these plants, or self-fertilization of these plants, right? Now when we do the self-fertilization, okay, so these are, now we are talking about the F2 generation, right? And in these F2 generation, the genotype is capital R, small r, capital Y, and small y, right? So let's take this as mother, okay? So what about the gametes? Now we have to form the gametes. So again, in formation of the gametes, I have to take one single character, that is capital R, and the other character, that is capital Y over here. Okay, so I'm getting a gamete which is having the two genes, and both are dominant genes, okay, that is round and yellow, right? Similarly, we have to make the gametes, other three gametes also, R and Y. Okay, here small r, capital Y, and here we are getting small r, small y, right? So we have the gametes which are egg, because these are produced by female. We have taken female, right? Because the male also, because we are doing the self-fertilization, so male also have the same type of genotype. So in case of male also, I'll be getting the sperm which are having the same type of genes present in the gametes. In case of male also, I'll be getting the similar type of gametes, RY, capital R, small y, right? Small r, capital Y, and small r, small y. That's it, right? Now what we have to do, so here we can arrange, the sperms are arranged over here and the eggs are arranged over here. Okay, these are the gametes which are formed from the male and the female, right? Since the chromosomes segregate independently at meiosis, the F1 progeny give rise to 4 different types of gametes, because these gametes are separating independently from each other, right? That is why we are getting 4 different type of gametes. What do we mean that? It means that these gametes are not moving from one another in a package. Suppose capital R and capital Y, they are not moving together to form the gametes. They are getting separated. As you can see, the capital R first mixed with Y, first get paired with Y, then this capital R get paired with small y. So capital R and capital Y, they are not acting like a package. So every gene which is present on the chromosomes, they are separating independently from each other, and this law is known as law of independent assortment. Okay, now let's see the result of Punnett's question. Now here we have already arranged the sperms and egg, and when we do the when we do the crossover, we will find that capital RY and capital RY, first they will come, and we are getting round and yellow, right? Round and yellow because capital R is there, which is responsible for the round character. Capital Y is there, which is responsible for the yellow character. Okay, so wherever you found capital R, which is dominant character, capital Y, which is dominant character, you will find these trait, that is round and yellow. So there is one round and yellow over here, one, and the another round and yellow, as you can see the genotype. Okay, here also another round and yellow, though small r is there, but that is recessive trait. So this is the third one, and again round and yellow. This is again the fourth one. Capital R, capital Y, definitely round and yellow. And here capital R, capital Y, fifth case, and this is not the case. Here also capital R, capital Y, this is the sixth case. Then here also seventh case and eighth case and ninth case. Okay, wherever you see capital R, capital Y, that means round and yellow type of seeds is produced. So for round and yellow, okay, for round and yellow, 9. Okay, out of 16, a round and yellow, 9 round and yellow seeds are formed. What about the other characters? Round and yellow we have taken. Now we are going to take round and green, right? I'll make it capital, round and green. Fine. So round and green, as we can see here, there is a R, capital R, round is there, but no capital Y. That is why it is round and green. Here also, this is the one round and green. Here capital R is there, no capital Y, so round and green. Second case, and here you can see capital R, no capital Y, again the third case of round and green. Okay, and this is what we got, round and green. Now coming to the wrinkle. Okay, so we will come to the wrinkle and small r, wrinkle and yellow. So yellow is a capital Y. So if you see wrinkle and yellow is a one case, wrinkle and yellow. Here is the next case, wrinkle and yellow, and the next case, wrinkle and yellow. So altogether wrinkle and yellow, 3 plants are found. Okay, and what about wrinkle and green? So small r stands for wrinkle and small y stands for green. So there is one case of wrinkle and green. So the phenotype ratio is 9 is to 3 is to 3 is to 1, which is very important from the point of view of exams. Here you can see the ratio 9 is to 3 is to 3 is to 1, and he took so many seeds. He took 556 seeds and he found these many plants, and the approximate ratio he got is 9 is to 3 is to 3 is to 1. So we couldn't find the genotype. The genotype is very complicated, so only thing you have to learn is the phenotype of the F2 generation of dihybrid cross. Now let's see the observation of Mendel's dihybrid cross. What we have observed in the first filial generation, we have observed that all the plants were having round and yellow seeds. In the second filial generation, for his surprise, he got 3 different type of combination of seeds, that is round and green, wrinkle and yellow, and wrinkle and green. And that is why he got the ratio 9:3:3:1. So what he concluded from his experiment, that round and yellow are dominant character. Okay, because these two characters were expressed in F1 generation. Okay, these two characters were expressed in F1 generation. If you have seen the parents in the F1 generation, they were having round and yellow genes which are dominant, and wrinkle and yellow genes which were recessive. But at the end of the F2 generation, that is as the result of F2 generation, we came to know the other three combinations also. Okay, so which is possible only in the heterozygous condition. Now, the second point, occurrence of the new phenotype combination shows that genes for round and yellow seeds are inherited independently of each other, right? We never got, in the F2 generation, there were only 9 out of 16 seeds, 9 were round and yellow. Okay, if round and yellow always goes in package, if they are not going independently into the different genes, then I will always get this combination of round and yellow, right? So this shows that the genes, okay, were inherited independently. They move into the gametes independently, right? This is known as law of independent assortment. Now let's read this law. Alleles of different characters separate independently from each other during the gamete formation. Okay, I have showed you the formation of the gamete. R was combined with Y, right? Then again capital R was combined with small y, right? And this is how we made gametes, right? So independently the gene for the round character was combining with the other genes, right, of different characters, right? So this is known as independent assortment. Now coming to the mechanism of inheritance, which is very important to learn, and this is the conceptual uh, based, or we can say this is the concept of inheritance, right? So let's start with the first concept. Here each gene set is present not as a single long thread of DNA, but as a separate independent piece, okay, independence piece, each called a chromosome. So here in the picture we can see that there is a cell. Inside the cell there is a nucleus, and inside the nucleus you can see the chromosomes, right? Now if I uncoil this chromosome, I can see the DNA structure, because we know that the chromosome is made up of DNA. On that DNA you can see a segment of DNA which we called as a gene. And what is gene? What is a chemical composition of gene? Chemical composition of gene is nothing but it is a protein, right? It is a special type of protein responsible for a particular trait, right? We have done so many traits. We have done round, yellow, green, wrinkle, tall, short. These all are traits. So these traits are controlled by a special type of protein which we call gene, which is basically a hereditary unit in organism, right? So here you can see a protein, and these are present in the chromosome. This is what they want to this is what the statements wants to tell you, right? Then each cell gets 2 copies of chromosome, one from each parent. So we know that the chromosome, let's take a chromosome over here. This chromosome is a chromosome which which a progeny got from his father, okay, this one. And the other chromosome, I'll change the color. and this is the another chromosome which progeny got from his mother, right? So altogether every cell like this, every cell will have two sets of a chromosome uh, which the progeny is getting from his or her parents, right? Okay, it can be maternal or paternal. That is from father. One the progeny is getting from father, the other chromosome the progeny is getting from mother. Together, these two chromosomes having the same shape and size, we call them homologous chromosomes. Clear? Then each germ cell or gametes has one copy of it. Now if this is a parent, okay, when this parent or let's say when this boy gets mature and the sperms are getting formed in their reproductive organs, then we know that sperms are nothing but gametes. Then each set of this chromosome will enter in a gametes. This gamete and the other gamete will be having the other chromosome, right? Here you can see these chromosomes are getting separated. Okay, these chromosomes are getting separated in the gametes, and we know that in meiosis four gametes are formed. Just because I have taken the one set of chromosome over here, so I'm showing you how these chromosomes are going inside the gametes. That is one gamete from one uh, chromosome from father will go to one gamete, and in another gamete the different chromosome which a progeny got from his mother will enter. Okay, so this is what is happening. So each germ cell or the gamete has one copy of it because there is a reductional division in the sex organ at the time of formation of gametes, right? So if I say this is a 2n, that is diploid condition, then this is a haploid condition. What is a haploid condition? Half the number of chromosomes, right? I'm not showing here all the 23 number of chromosomes, but I'm showing only one set of chromosome. This is two chromosome over here. When the formation of gametes, okay, there are only one set of chromosomes, that is half the number of chromosomes. Okay, so in human beings we have 46 number of chromosomes in the individual, but in the gamete cell, in the gametes, there will be 23 number of chromosome. That is half the number of the somatic chromosome, or the chromosome which are present in the body cell. Then when fertilization takes place, normal number of chromosomes is restored in the progeny, ensuring the stability of DNA of the copies. Okay, so here in this picture they have showed the diploid number. You can see this homologous chromosome. There are three homologous chromosomes present over here, right? In this three homologous chromosome, this one may be coming from father, the other one coming from mother. Okay, here also one coming from father and one coming from mother. Clear? Here also one coming from father and one coming from mother. Okay, while when the gametes are formed, here the gametes, these are the gametes, right? When the gametes are formed, let's take what are the gametes over here. Gametes over here can be eggs, or they can be sperm, right? Egg or sperms. TES, children. So we can see here there is only one chromosome is entering inside a gamete. Only one chromosome out of the two chromosome, only one chromosome is present in the gamete over here, which is responsible for a set of traits. Okay, so these are the haploid number. So how many are there in diploid? Over here there were 6 chromosome. In haploid there are only 3 chromosomes. That is half the number of chromosomes. Okay, so why this is possible? Because we know that, let's take the sperm, which is having 23 chromosome in it, and let's take the egg, okay, which is having 23 chromosome, right? That is n number of chromosome or haploid number of chromosome. When they fuses together, the 46 number of chromosome will going to retain in the progeny. Okay, if this wouldn't happen, then definitely 46 and 46 92 number of chromosomes will be present in the progeny in the one generation. Then again 92 plus 92, this is how these chromosome number will increase every time, and you will see a different organism every time, right? A different species probably every time. So to maintain uh, the similarities between the parent and the basic similarities between the parent and the progeny, these number of chromosomes should be maintained. Now let's take the another topic and the last topic of this chapter, sex determination, right? Okay, now how is the sex of a newborn individual can be determined? Different species use very different strategies for this. Some rely entirely on the environmental cues. Thus, in some animals, like a few reptiles, the temperature at which fertilized eggs are kept determines whether the animal developing in the egg will be the male or female. So, in other animals, such as snails, individuals can change sex, indicating that sex is not genetically determined. However, in human beings, the sex of the individual is largely genetically determined, right? So, in other words, the genes inherited from our parents decide whether we will be boy or girl, right? But so far, we have assumed that similar gene sets are inherited from both parents. If that is the case, how can genetical inheritance determines the sex? So, okay, so let's see the explanation of this. Okay, so we know that the chromosomes, these are the last pair of the chromosomes which are responsible for the sex of an individual. Okay, so if a male is carrying X and Y, okay, genes, and female, we know that female carries X and X genes. Female has only one type of genes, that is X and X, or we can say female has only one type of chromosomes, that is X chromosome and X chromosome, whereas males have X and Y chromosome, right? If I cross over these X and Y chromosomes, so I'm getting the first female child, and if I just cross over here, I'll put a crossover sign, right? And here I'll get male, right? And here again I'm getting X and X, and the other here I'm getting X and Y, which shows that there are 50-50% chances. So 50 and 50, that is 50% chances of male and 50% chances of a female boy. Okay, by now you must have come to know that when a progeny has X and X chromosome, this progeny will be a female child, right? And when a progeny has X and Y chromosome, this will be a male child, right? So let's read out the statement from here. The process of determining the sex of an individual based on the composition of genetic material is called sex determination, right? In different animals, sex of an embryo is determined by different factors that we have discussed here. In humans, sex determination happens on the basis of presence of Y chromosome, right? Only on the presence of Y chromosomes, okay? So XX is a female, XY is a male, and ovum always contain, ovum, female egg ovum always contain X chromosome, okay? And definitely it will have one X chromosome. We know there are 4 gametes formed after the ovulation, and all these gametes are having X chromosome in it, right? So eggs will always have X chromosome, ovum will always have X chromosome, whereas in the sperm there are different chances. Sperms can have X chromosomes and sperms can have Y chromosome, okay? And if a X chromosome sperm fuses with the X, okay, then the progeny, then the child will be a female. Fine, it will be a female. So I'll represent it female by this. And when a Y chromosome carrying sperm fuses with the egg, then the newborn baby will be a boy. So this is how we determine sex in human beings. We know that sex chromosome in human beings is the last pair of chromosome. Here you can see there are 23 pairs of chromosome, right? This is a karyotype of a human being. Out of these, 22 pairs of chromosomes are called autosomes, and the last chromosome is known as sex chromosome, right? If a person has XX chromosome, then she will be a female, and if he has XY chromosome, definitely he will be a male. Okay, this possibility I have already discussed with you. This is based on the Mendel's law. Okay, this shows that half the children will be boy and half the children will be girl. So 50% probability is there for male and 50% probability is there for female. Okay, all children will inherit its X chromosome from their mother regardless whether they are boy or girl. Okay, thus the sex of the children will be determined by what they inherit from his father. This also we have discussed, and not from their mother. Okay, so this is all about today's topic, children. Thank you so much, and practice today's topic.